Assalamualaikum warahmatullahi wabarakatuh. Our journal is the impact of nutrition and environmental epigenetics on human health and diseases. Epigenetics is the control of gene expression without direct changes in the DNA coding sequences. It will modulate and regulate gene expression through various epigenetic marks, which is the chemical compounds added to DNA or histone proteins, and it will prevent the binding of transcription factors to DNA or by allowing the binding and upregulate cellular processes. The exposure of environments will affect the epigenetic marks. Environmental exposure like environmental factors or the lifestyle including the behavior, nutrition and toxins will change the epigenetics. Environmental exposures like environmental factors, lifestyle including behavior, nutrition and toxins will affect epigenetic changes, and evidence suggested that many adult diseases including cancer have epigenetic origins.
Nutritional epigenetics. Nutritional epigenetics, the future. Diet, a nutrient, has shown to reversibly alter the DNA methylation pattern, histone modification and chromatin remodeling, and in addition shown to alleviate the aberrant DNA methylation or histone acetylation from previous diet deficient status. This has ultimately further altered gene expression, leaving an impact on overall health. Furthermore, it is noted that the diets in early life plays a critical role in determining DNA methylation. The epigenetic effects of nutrition in the gestational period induces long-term changes and in general expression as the modification in DNA methylation or histone acetylation. These epigenetic modifications, as attributed to the specific bioactive food components, have altogether resulted in the possible making of a personalized nutrition intervention.
Nutritional epigenetics and cancer. Folic acid and vitamin B12 are two examples of epigenetically active ingredients that play important roles in DNA metabolism and also the DNA methylation. In one study, folic intake is positively correlated with P16 tumor suppressor gene. So low intake of folate has been linked to hypomethylation and increased risk of colorectal and pancreatic cancer. Folate deficiency reduces the potential for DNA methylation and abnormal DNA methylation which is associated with many types of cancer. Another potential epigenetic effects of nutritional component is antioxidant. Diet rich in fruits and vegetables containing antioxidant can protect against cancer. So as an example, green tea that contain polyphenol which have shown reduction in DNA hypermethylation of critical genes resulting in tumor suppression. So epigenetic roles against cancer. This is a summary of some dietary components considered to have protective effects against cancer. The first one is the methionine nutrient. It could be found from the sesame seed, Brazil nut, fish, and spinach and peppers. The epigenetic role is important in the S-adenosyl methionine synthesis, which is used for polyamine synthesis and essential for cell proliferation. The other nutrients are folic acid, vitamin B12, and vitamin B6, that their epigenetic role is in the methionine synthesis that plays a critical role in the metabolism and also major components in the cellular metabolism. There are several other nutrients with different of their epigenetic roles. Transgenerational effects. Diets are really important as it does not only affect the epigenome to modify the individual but also the transgenerational phenotypes. So the first one is the Western diet. Western diet is a diet high in saturated fats, sodium and sugar. This diet has been linked to many diseases include hypertension, heart disease, diabetes and obesity. Generally, it also been linked to an increased risk of cancer. So the individual effect is it produces the obese phenotype, which could lead to fatty liver disease, cancer, and also immune disorder. and the transgenerational effect that they produce is the disease phenotype, which will lead to cancer, growth abnormalities, increased risk of metabolic disorder, and also type 2 diabetes. It is also reported that in one study, maternal dyslipidemia caused significant epigenetic changes in placentas and fetal liver, and also increased the fetal liver triacylglycerol accumulation. And the second one is the Mediterranean diet or the healthy diet with a reduced risk of heart disease and cardiovascular mortality. The Mediterranean diet traditionally includes fruits, vegetables, pasta, rice, fish, and healthy fats, which are monounsaturated fats and polyunsaturated fat. So the individual effect is the lean phenotype with low disease risk and delayed aging. The transgenerational effect is the normal phenotype with reduced risk of disease. So the Mediterranean diet is also associated with a reduced incidence of cancer and the risk of breast cancer, because it has been reported that the reduction was found on women who eat a Mediterranean diet supplemented with extra virgin oil and mixed nuts.
Nutritional epigenetic in health and disease. Nutrition is one of the environmental epigenetic factors, and in a study to observe the association of prenatal nutritional conditions in postnatal health, where it observed the effect of starvation during pregnancy, the outcomes is it increased the risk of diseases. The second is the transgenerational effects of poor maternal diets, where the grandchildren of the woman exposed to famine or other dietary alterations through pregnancy will experience health complications. The molecular mechanism is the methylation of spermatozoa nucleus, where it is further transmitted by paternal lineage, where methylation of gametes occur. The third one is the methyl deficient diet. It decreases DNA methylation but reversible. However, in diets high in methyl-donating nutrients, it can rapidly impact gene expression, like folate or choline. It will permanent hypomethylation. It will cause permanent hypomethylation of parts of the genome.
Endocrine disrupting chemicals, EDCs, are exogenous agents of environmental chemicals that mimic natural hormones by interfering the hormone signaling pathway, which is critical for many processes involved within the system itself. The EDCs can include its global effects as its effect on enzyme like DNMT expression. And the EDCs also can include its gene-specific targeting action, which are the genes targeted are responsible for recruitment of histone modifiers and maintenance of DNA methylation status via its interference with DNMT. It can also further regulate other non-coding RNA expression, like microRNA. EDC exposure induces abnormal development, behavior, and disease. This includes environmental epigenetics, prenatal EDCs exposures. EDCs. The interference of EDCs to hormone signaling induction has caused significant hormonal imbalance and resulted in several disorders manifestations. This includes the increasing development of tumor, which is tumorigenesis in children and adults after fetal exposure. The epigenetic changes can transfer over generation as the implicated epigenetic modification, including the non-exposed offsprings. The prenatal EDC's exposure has been coherently related with BPA doses deficit level of exposure, which has ultimately predisposed to various sexual, developmental, and behavioral disorders. At very low BPA level, the sexual disorders can include the impaired semen quality and timing of puberty following testosterone or any hormone-related imbalance synthesis. The development aspect is also affected via its cognitive aspect that can lead to learning difficulties. Very low BPA exposure also can cause behavioral changes, leading to several issues like hyperactive disorder.
EDCs, epigenetics and cancer. For the introduction, it is important to remember the effects of EDC vary based on different doses, different ages and different target tissues. For prenatal DES exposure, it can lead to many events, such as uterine tumor. For genistein, exposure can alter mammary gland structures and increase risk of breast cancer, while for dioxin, it is a developmental and reproductive toxin. PAH exposure can lead to many events, such as lowered immune functions, increased risk of cancers, poor neurodevelopment, and many more. Other toxin exposures and their epigenetic effects. Pre-natal smoking will cause epigenetic modifications in buccal mucosa cells, will cause health, neurodevelopment, behavior, cognitive functions to be affected. Maternal tobacco smoking during pregnancy will mainly induce respiratory diseases associated with the immune system and also cancers. For alcohol, it can be risk factors for various cancers, deleterious or hazardous effects to growth, metabolism, and neurodevelopment, and also if it will impact particular memory and learning process, malformations, abnormal fetal development. What are the effects of environmental epigenetics towards cancer, for example, breast cancer? For MSC and TCS exposure will lead to ovarian cancer. For dioxin, it will cause delayed mammary gland proliferation and differentiation during puberty and delayed breast development in females. For atrazine, it will cause delayed early mammary gland development in female offspring and prolonged sensitivity to carcinogens. For BPA, it will lead to alteration in both male and female mammary gland morphogenesis and later may lead to breast cancer. These are the mechanisms. For vinclozolin exposure, it will alter structure of mammary gland and increase risk of breast cancer. The highlight here is there is correlation between exposure to estrogens, phytoestrogens, and high-fat diets, and breast cancer susceptibility is significant.
We will now move on to the potential of epigenetic drugs. Histone deacetylase HDAC and histone deacetylase inhibitors are the two potential of epigenetic drugs. For histone deacetylase or HDAC, there are four classes of drugs. For HDAC class 1, the enzymes are expressed in nucleus. This applies for HDAC 1, 2, 3, and 8. For the second class, HDAC class 2A, the enzymes are expressed in cytoplasm and nucleus. HDAC 4, 5, 7, and 9 falls under this category. For HDAC class 2B, the enzymes are expressed in cytoplasm, and this applies for HDAC 6 and 10. The last class, HDAC class 4, has only one drug, which is HDAC 11. For histone deacetylase inhibitors, the therapeutic mechanism for this drug is by helping re-establish the epigenetic control, inducing the growth arrest, differentiation, apoptosis in malignant cells, and by inhibiting enzymes responsible for the transcriptional repression and silencing. This will reverse the gene repression observed in the malignancies. There are two types of drugs that can be used as an example for histone deacetylase inhibitors. The first drug is the vorinostat. It is normally used to treat relapsed or refractory cutaneous T-cell lymphoma, or CTCL. It also causes cell cycle arrest and cell death. For the second drug, which is romidapsine, it is an anti-cancer chemotherapy drug. It is used to treat cutaneous T-cell lymphoma, CTCL, and peripheral T-cell lymphoma, PTCL. It blocks the HDACs, so histone cannot be used properly, which will interfere with the genetic makeup of cancer cells, and then leads to cell death. Currently, the research that are done on the epigenetic drugs can be seen in two research. The first one is by the usage of the drugs to reverse immunosuppression and restore T-cell mediated anti-tumor activity. The second research focus on how the epigenetic drugs acts as an immunomodulatory agent in treatment of cutaneous T-cell lymphoma. We will now move on to the conclusion. In conclusion, Individual health depends on interaction of many environmental factors with its genetics. The second conclusion is that lifestyle factors, be it nutrition, behavior, environmental toxin exposure, it can lead to the epigenetic mechanism. The last one is that the factors may lead to epigenomic alterations and the changes in epigenetic marks which will impact the gene expression. What we can learn from this paper is that understanding the molecular mechanism and signaling pathways in environmental epigenetics, it will lead to the reduction of societal impact on the public health. The second is that nutrition and chemical pollutants are two important environmental factors which will affect human health. These have a direct impact on individuals, which contribute to the pathogenesis of various diseases such as cancer. The factors that lead to the epigenetic transmission, be it hereditary, it can lead to a major global public health issue for the future society.